Okay. So, good afternoon, everyone. Thank you all for being here today. So during today's presentation entitled Genetics and Genomics of Rose Resistance to Black Spot Disease, I will intend to provide an overview of the work carried out the past 3 years and a half in the Institute of Research in Horticulture and Seeds. This thesis was conducted under the supervision of Fabrice and as a PhD director and Vanessa and Laurence as co-advisors. I would like to thank all the members of the jury for accepting to review this thesis. So as a general introduction, I will try to provide some background elements on the challenges that rose industry is facing nowadays, as well as on the pathosystem that I studied during my research. It can be affirmed that roses are the most important flowering plant worldwide, and this for centuries now. But what are roses? The roses, we all know, belong to the family Rosaceae and the genus Rosa. Rose classification is very complicated, but what you have to remember today is that there are many species and hybrids in this genus, but just a few of them have been used over the years to breed new rose cultivars. There are two centers of diversification that remain isolated until the 1800s. After that, the Asian cultivars were introduced into the European pool, and this gave birth to the modern roses we all know today. There has been an intensive selection and breeding over the centuries with intercrosses between rose species and cultivated forms. The result is a development of thousands of cultivars with different flower shape, color, size, and growth types, for example. So nowadays, rose represents the most important ornamental plants worldwide. There are three main outlets in rose industry. The cut roses that dominate the world trade of cut flowers. Then you have the potted roses. It is a new niche market that has significantly increased over the last years. And the third is the garden rose group that has a wide variety of cultivars with different shapes and colors. Finally, we can mention the marginal but important use of roses that is for fragrance, essential oils, and even food industry in some countries. So for the presentation of today, I will focus on garden roses. In the past years, a loss of attractiveness has been observed in garden roses. It is observed in several countries. Indeed, roses face many threats nowadays. And so first, let's talk about the pathogen threat. As beautiful as roses are, they have to face many natural enemies, from pests to fungal disease. And because there are so many of them, I will only focus on the fungal disease attacking leaves. The most important ones are Cercospora, rust, powdery mildew, and black spots. So among the diseases previously mentioned, black spot is the most serious disease of landscape roses in warm, humid zones. This disease occurs mostly in gardens as well as public spaces in the northern and southern hemisphere. This global spread and close proximity with the population has made it the most well-recognized disease worldwide. So the disease starts affecting the lower part of the rose bushes, and the typical symptoms are these black spots with fringe margins that appears on the upper side of the leaves. The disease spreads from the bottom to the top, and the infected leaves start yellowing and falling, leaving only the canes and the flowers. This premature fall can be observed during the summer, and in the most susceptible cases, black spot can lead to the death of the plant. So the causal agent of this disease is Diplocarpum rosae for its perfect stage and Marsanina rosae for its imperfect stage. It is a subcuticular fungus that grows right under the cuticle, and it has a very interesting lifestyle as it is an hemibiotroph that feeds from live cells with the use of Hostoria, and then switches to a necrotrophic stage and starts feeding on dead material using intracellular IVs. So far, 13 races have been described, which denotes a large diversity in the fungal population. So this disease is present in epidemic proportions as the pathogen is capable of completing several cycles during the infection cycle window from spring to autumn. The propagation by water splashes increases its incidence at every rain of the season. This pathogen is particularly difficult to get rid of as it overwinters in plant material like dormant stems, prickles, and fallen leaves. For now, the most efficient way to control black spot disease is the use of fungicides with applications as often as 1 per week or 1 every 2 weeks. On the other hand, genetic resistance is more and more considered in disease management. So consumers' demands has shifted over the recent years because of the added cost of fungicide sprays, the health and environment hazard that it represents, but also the fact that it contributes to pesticide-dependent resistance. And so there are many reasons why consumers are no longer willing to buy pesticide-dependent landscape, but these are the most important ones. Therefore, disease resistance has been ranked the most desirable threat the last 10 years. Moreover, in Europe in particular, the restrictions in phytochemical use has been another threat that the rose industry is now facing. So for example, in France, the Ecophyto plan aims to reduce the chemical use in agriculture by 50% before 2025. And so in this context, several laws have been voted, and the use of phytochemical has been forbidden in public places and private gardens in 2017 and 2019, respectively. So there are many ways to reduce the incidence of the disease directly in the gardens without using the chemicals. But to solve the problem, one of them is the use of resistant varieties. So how resistant are the roses we know? So two studies demonstrated that in average, less than 8% of the commercialized varieties presented resistance to black spot disease. While in the wild roses, 30% of them presented this resistance. So these results point a lack of resistance in the available cultivated pool. However, after the 1900s, many research programs have been working together with breeders on black spot disease resistance and have been developing varieties with higher degrees of resistance by using natural resistance present in the roses. So an improvement of black spot disease resistance is expected in the future and can be already observed in the newly released varieties. So to improve disease resistance in roses, many studies need to be carried out. So what is genetic resistance? There is a wide variety of terms that have been used throughout the years. This diversity clearly reflects the interest of researchers and all the assumptions made to explain this complex concept. Indeed, in countless publications, these terms have been used and opposed to each other in a strict dichotomy that simplifies greatly the reality in disease resistance. Over the years, scientists have used and misused these terms by treating them like they are synonyms and by making assumptions about the relationship between the phenotype, the interaction type between the host and the pathogen, and the genetic basis. So in the list you have here, these words refer mostly to the phenotype, these ones to the interaction type, and the last ones here to the genetic basis. So before talking about what we know on the genetic resistance to black spot disease, I would like to present the rose genome and its complexity. Rose has a base of 7 chromosomes. The genus is an interspecific complex with genotypes presenting different levels of ploidy, from diploids to decaploids. Rose are highly heterozygous, and when crossed, they show a strong segregation distortion due to incompatibilities. During my PhD, 2 sequences were released from Rosa chinensis Old Blush, but because of roses being very heterozygous, double haploid lines were used to build these 2 genomes of high quality. The genome size was estimated at 512 megabases. So now, what do we know about black spot disease? There are 2 main types of resistance that have been described in rose genotypes. On one hand, there is a complete resistance for which no sporulation can be observed, and the pathogen development is severely restricted. This type of resistance has been described in genotypes like Rosa multiflora, George Vancouver, or Bright Eyes. On the other hand, you have the incomplete and partial resistance that allows the pathogen to grow, but at a limited extent, and for which some sporulation can be observed. This type of resistance was described in Rosa bichurana, Applejack, or Ballerina, for example. So extensive work has been carried out to identify the genes underlying complete resistance in several genotypes. 4 major genes have been described so far, and these genes have been shown to have a race specificity with different degrees of spectrum. On the other hand, studies on incomplete resistance remained limited. Partial resistance has been shown to segregate quantitatively in the Applejack variety. It can also be described as race specific in some cases, but also race non-specific in other cases. So partial resistance in black spot disease seems to be highly complex and dependent on the genotype that we consider. Finally, the existence of a significant general combining ability and a strong additive genetic effect has shown us that it is possible to integrate partial resistance in the new release varieties. So the problem is that an important deployment of the same disease resistance, even in different varieties, can apply a strong selective pressure on the pathogen populations, so it can select virulence alleles that can bypass the resistance. So using incomplete resistance in roses can be one solution to alleviate the selection pressure on the pathogen population. But to be able to do that, more studies need to be carried out in roses to identify the genes responsible for incomplete resistance. So during my PhD, I focused on the study of the partial resistance present in the rose pool. So let me present to you the research objective and the strategies that we implemented in order to fulfill them. The main objective of my PhD was to identify and characterize new genomic regions evolved in the resistance to black spot disease. We use a hybrid of Rosa vishurana. As in previous studies conducted in our team, this genotype has been evaluated with a good field resistance. Members of the Rosa vishurana species have been reported to exhibit a field resistance. Moreover, this species has always been referred to as partial resistant in the literature. So, in order to do that, we proposed a multi-scale study of the partial resistance in RW. First of all, we wanted to study the resistance in different populations sharing a common parent during a multi-environment and multi-population QTL analysis. Then we wanted to focus on what happened at the individual level directly on the leaves to perform a thorough phenotypic and microscopic study of the pathosystem. Finally, we wanted to investigate the main pathways that are activated during the resistance of the genotype RW. So today, and to better characterize the partial resistance of RW against black spot disease, I propose you to build the identity card of the partial resistance present in RW. In this first part, I will briefly present the results of the QTL study that was published in the TAG journal. Several QTL detections have already been carried out in roses using many different maps with FLP, SSR, and MBS markers. Recently, with the development of the SNP array, high-density genetic maps were built for diploid and tetraploid populations. In addition, due to strong inbreeding expression and highly heterozygosity, the outbreak crosses are used to develop F1 populations, and therefore the double pseudotest cross strategy is applied to perform QTL analysis in roses. This way, two separated genetic maps are considered with the markers only heterozygous on the male parent or only on the male parent or only on the female parent. So in our study, we used 3 populations connected by the male resistant parent RW, and this resistant parent was crossed with H190, creating HW that was planted in 3 locations in France. Then RW was crossed with 2 other genotypes, a susceptible genotype oil blush that created the OW population, and the resistant genotype Zephyri that created the FW populations. These 2 populations were planted in Angers. So all 3 populations were scored between 2012 and 2018 for all the years. Natural infections in the fields were used to evaluate the population. So black spot disease was evaluated on the lower part of the plant using a rating scale from 0 to 5 that correspond to the percent of infected leaves and also to the defoliation degree. So 0 represents no infection observed and 5 a complete defoliation, for example. So like explained earlier, a double test cross strategy was used and the maps were already published for FW and HW. These maps were built with SSR markers, whereas the map used for OW only had SNP markers. So during my PhD, I added common markers that were found between all these three populations in order to connect these three populations. Then we perform an appropriate QT analysis according to the distribution of the phenotype. So for the normal distribution or close to normality, we used a normal model. And for distributions presenting a spike-like, so either at the beginning of the distribution or at the end, we used the two-part model that was described implemented in the package RQTL. Finally, we use a meta-analysis to integrate all the individual QTLs that were detected over the years and the locations. And all these individual QTLs were combined in order to find the most likely location that is linked to black spot disease resistance. So as we are focusing on the partial resistance of RW, I will only present the results that we found on the male maps. So here, in this case, we found two main QTLs on linkage group B3 and B5, and for all populations, years and locations. Also, we identified two QTLs on B4 and B6 that were background dependent. Then the QTLs found on B3 explained 4%, between 4% and 24% of the phenotypic variance, while the ones used on B5, the one found on B5, explained between 5 and 20% of the phenotypic variance. Then we were able to better characterize the QTLs detected after using the two-part model for the distributions that were non-normal. So two-part model, like its name indicates, divides the traits in two parts: the binary part that studies the presence, absence of the disease, and then the normal part that analyzes the score above or under the peak. So either here 0 or 4. So during the QTL analysis, two loss scores are calculated. So the first loss score that shows the penetrance here in blue, and the second loss score that shows the an effect of the QTL on the severity that is here in green. So we perform this analysis for the scoring years for FW and HW. And we saw that the QTLs found on B3 affected both the penetrance and the severity, while the ones detected on B5 only affected the penetrance. Then we wanted to reduce the confidence interval of all the QTLs detected individually over the years. And so here in this figure, you can see all the individual QTLs that were detected for the linkage group B3 and their corresponding confidence interval. And with the meta-analysis, we detected two meta-QTLs at the beginning and at the end of the chromosome. The color you can see here correspond to the contributions of each QTL to the meta-QTLs that were calculated. So for linkage group B5, we can see here that the QTLs found on OW contribute to the first meta-QTL, whereas the rest of the QTLs that were found on all the other populations contributed to the second meta-QTL. Only 1 year of OW was found to contribute to the second meta-QTL, but this scoring year showed a lot score with 2 peaks. And the first one, we can see that it colocalized with the first meta-QTL, and the second one colocalized with the second meta-QTL. So we can see here that there are 2 meta-QTLs that were detected for each linkage group. but their existence remain unclear, and we need to do more analysis for that. An interesting fact is that this second meta-QTL colloquialized with the markers found to be close to RDR4 major gene that was found in the bright eyes genotype. So far, we have seen that the partial resistance of RW segregates quantitatively, and that new insights on the genetic basis were found with the description of two main QTLs on B3 and B5, but also with the descriptions of two QTLs that were background dependent, so on B4 and B6. Now, let's go to the second part, and I would like to present the studies that we carried out to better characterize the partial resistance in black spot at the phenotypic and microscopic level. First, some further information on the development of Diplocarpon rosae under the cuticle. Here you have an overview of the pathogens infection from Gashomo and al. Study. And from their observations, I was able to create the following diagram to summarize the pathogen's development. So the pathogen's conidia needs free water to germinate, and it penetrates the cuticle, mediating an apressoria or not. Then several structures will develop under the cuticle, the first ostoria can be produced within several hours. Then long distance hyphae are developed and they will grow gradually from the entrance point to colonize remote areas of the leaf. And then further growth can be observed in the lower part of the leaf mesophyll, with the production of short distance hyphae. The timing of all these steps depends. Then the leaves show to have some necrotic cells that appear. And finally, the pathogen develop disacervally that will release the new produced conidia. So the timing of all these steps depend on the genotype that we use and the strain as well. And so it is important to study the pathogen cycle with the genotype that we used. So two types of interaction between Diplocarpon rosae and the host can be described. A compatible interaction where the fungus is able to complete its infection cycle, and incompatible interactions that denotes a lack of development of this pathogen on the host. So for the first one, 5 types can be denoted from the most susceptible one to the least susceptible one, and for the incompatible one, from the more susceptible to from the least susceptible to the most susceptible one. So for this part, I will focus on the battlefield of partial resistance. So first, we wanted to identify the time points corresponding to the steps of Diplocarpon infection. And we used the whole plant assay for this study. And then we wanted to see if RW interaction with Diplocarpon is different from the ones that can be observed for RDR carrying genotypes. And for this, we used the detached leaf assay. And we also saw with preliminary studies that there seems to be a leaf age effect on RW partial resistance. And so this study was carried out on both whole plant and detached leaf assay. The whole plant assay was carried out in France in my team, and the detached leaf assay was carried out in Hanover in Professor Deppner's team. So for the whole plant assay, we used Old Blush and RW. So Old Blush as a susceptible genotype and RW as a partially resistant. So cuttings were inoculated in the greenhouse using the DIFRA 67 strain, and mock inoculations were also performed. The first fully developed leaf were also marked in order to study the leaf age effect. Then, after the inoculations, the plants were covered for 3 days, and then we sampled at different time points from 0 to 16 days, and samples were processed and then stained with aniline blue to see the pathogen development and the plant's reaction. Then at 16 days, we assessed the disease on whole plants. For the disease assessments, we decided to divide the leaves between young leaves that denotes the group 1, and these young leaves are still growing, and old leaves that denotes the group 2, and these leaves have stopped their growth. So for the detached leaf assay, we used again RW and O blush by using old leaves and young leaves. And then we used new genotypes, so Pariser charm as a susceptible genotype. And then we also use Pariser charm carrying RDR1 gene, a transgenic Pariser charm carrying RDR1 gene, as well as two RDR carrying genotypes, so George Vancouver and Bright Eyes. And finally, a member of the Rosa maialis species that has been shown to be very resistant to black spot. So for these genotypes, we only used young leaves. These genotypes were inoculated with DIFRA67, and again, a mock inoculation was performed. Several drops were deposited on the detached leaves, and the samples were covered, and the drops were removed after 3 days. And after 9 days, we sampled all the genotypes, but also at 23 days for only the resistant genotypes. The samples were stained with a wheat germ agglutinin associated with Alexa fluor 488 to stain the fungal structures, and with an aniline blue to see the pathogens, the plant's reactions. So first of all, the study of the infection cycle allowed us to see that the germination happened at 2 DPIs on the susceptible genotype O blush, and that the entry happens at 3 DPIs. The IFA growth was observed around 7 DPI, and then the acervical formation started at 9, and the conidia was released at 16 days. For the resistant genotype, we were able to observe the deposition of callus at 5 days. And so we decided to use the following time points, so 0, 3, 5, and 7 for the next for the next transcriptomic study. So then we assess different components of the partial resistance in the leaves with different ages. For example, here we studied the percentage of the leaf that is infected, and this was done on OB, on O blush, and Rosa viscerana. So here you can see that the percent of the leaf that is infected is similar in RW young leaves, is similar to the one observed for the susceptible genotype. And then the older leaves didn't show a development of the pathogen. So then we looked at the percentage of the leaf that react with typical brown necrotic spots that were not observed for old blush, but that were only observed in RW. We can see here that younger leaves reacted more than older ones, and this denotes a reaction, a differential reaction between leaves of different ages. So looking closer, the size of these brown spots were different between old leaves and young ones. And you can see here that the necrotic spots are much wider and important in younger leaves. Then we observe both types of leaves at the microscopic level to better understand at which moment the infection is stopped. So here in the top pictures, you will see the results with the GFP filter in which we can see the fungal structures stained with the Alexa fluor. And on the bottom, you can see the results with the DAPI filter that revealed the aniline blue, so the plant reaction with callus deposition. So for old leaves, we observed that conidia germinated on the leaf and penetrated the cuticle. The cells, so we can also see that there is a deposition of callus around the penetration point, and also the cells surrounding the penetration point appear to be fluorescent. Then on the other side, you can see that there is 2 cases. So an early callus deposition that was similar to the one observed for the old leaves, and a late callus deposition that allow more fungal growth with long distance IFEs that were produced and Hostoria as well. So we can also observe that there is callus deposition in fluorescing cells in these young leaves, but the development of the pathogen showed that the resistance did not stop efficiently the pathogen at the beginning. So in addition, we investigated the type of interactions observed for different resistant genotypes, only using young leaves. And we observed that even if we only use young leaves, the resistance effectively stopped the pathogen development at very early stages. Whereas for RW, we observed that young leaves denoted more fungal growth than for the other genotypes, and that older leaves efficiently stopped the pathogens at the penetration point and did not allow the development of further fungal growth. So far, we have learned that the interaction type observed between RW and diplocarpal mosaic depends on the leaf age, with young leaves denoting a type 4 and older leaves denoting a type 7. We also saw that complete resistance observed in older leaves was efficient, and that younger leaves denoted an incomplete resistance. Early callus deposition was also observed in older leaves, and the localized cell death seemed to efficiently stop the pathogen growth. Now, we have seen what happens during the infection at the macroscopic and microscopic levels. It is natural to get a closer look to what happened and which mechanisms lead to the resistance observed in RW. Therefore, the last part, I will present the transcriptional analysis that we carried out. So several studies have described the defense response in roses against black spot disease, and two types can be mentioned here. The cuticle was observed as the physical efficient barrier, and then some secreted compounds have been shown to participate in the defense response of roses. So like phytoalexins, polyphenols, chitinases, and glucanases. Only a study on the defense responses in the compatible interaction were carried out using transcriptomics, so on the genotype Paris-Charm, and it has been shown that conserved but insufficient PTI response was characteristic of the defense response in this genotype, with PR proteins, chitinases, and defense-related transcription factors that were upregulated in this genotype. Then they also mentioned the existence of a partial resistance and salicylic acid signaling pathway seemed to be activated in the response to diplocarpone, but also the phenopropanoid and flavonoid pathways. So as we only have information on the defense response of a compatible, it is necessary to study the immune response in the case of an incompatible. Therefore, we investigated the response in our genotype RW that exhibits partial resistance. So to do this, we studied the incompatible interaction using the partially resistant RW and the susceptible one, O blush. So we only used old leaves of these genotypes, and we performed 3 independent inoculations using the strain DIFRA67, as well as mock inoculations for all the genotypes. Then for RNA extraction, we sampled at different time points that we mentioned earlier, so 0, 3, 5, and 7. And then we assessed the disease at 28 days using the scoring scale that we talked about in the part 1. So in order to study the transcriptome in a compatible and incompatible interaction, we conducted 2 studies. First, we performed pairwise comparison between mock and inoculated samples. And this for each time point and for each interaction type separately. So this denoted 8 comparisons. Then we studied the time course analysis for a single time series by separating the inoculated samples and the mock ones. So for the next part of my presentation, I will only present the results of RW as we are focusing on the immune response in the case of a partial resistance. So with the pairwise comparisons, we observed that a low amount of genes were found to be differentially expressed between inoculated and mock samples. And this was especially true for 0 and 3 DPI. as you can see here, with the upregulated genes and the downregulated ones. We saw also that with the PCA diagram here, that there is a good differentiation between the time points, with 0 being here, 3, and then 5 and 7 together down here. So we can see also that there is a strong variation between the biological replicates, which was expected as we perform independent inoculations on our samples. So seeing the lack of differentially expressed genes between inoculated and mock samples, we decided to separate the conditions between inoculated and mock samples. And we investigated the genes that were differentially expressed over the time for each condition separately. So for the inoculated samples, 3,000 genes were found to be differentially expressed over the time points, and they were clustered in different profiles, sharing the same expression over the time. And then for the mock samples, we found 3,900 genes that were differentially expressed over the time. And they were also clustered in different groups having similar profile of expression. So today I will focus on the comparison between group 4 and group 5 that shows an increased expression at 3 dpi and then a decrease in expression between 5 and 7 dpis. So for these two groups, we observed that the genes that were differentially expressed over the time were implicated in the defense response for many of them. And then we looked at the disease assessment of our plants after 28 days. And as expected, the inoculated plants showed development of disease for the all blush genotype, and then less disease development for the RW. Then looking at the read that mapped on the fungal genome of Diplocarpon rosae, we observed the same type of results, and so no contamination was observed on the genotypes that were not inoculated with black spots. So then we saw that there has been problems in our greenhouses with pardew mildew, and so we hypothesized that our samples might have been contaminated with pardew mildew. And therefore, we looked for the reads that mapped on the genome of Podosphaera xanthi. That was the only genome of Podosphaera that was available at that time. And we saw that there is a limited presence of pardew mildew in our samples, which can explain the presence of defense response genes in our mock samples. And so we hypothesized that inoculated samples had black spot and pardew mildew defense response that were activated, and that mock samples showed pardew mildew defense response activation. And so looking at the genes that were specifically expressed for the inoculated samples, we saw that there are genes that are involved in the response to hormone like brasinosteroids and salicylic acid. And interestingly, the GO-term defense response to incompatible interactions was significantly enriched, as well as the molecular function of transmembrane receptor protein. So when we look at the list of genes annotated with this GO-term, we observed that the majority of them were described to participate in the defense response, leading to hypersensitive responses. On the other hand, only 383 genes were found to be specifically differentially expressed in the mock samples. And we observed that GO-terms like HR and plant cell death were observed in these samples. And so all these results indicate that both samples both type of samples, so inoculated and mock ones, seem to have an activation of the HR response. However, it seems that RW activated different pathways to fight against black spot and pardew mildew infections. Finally, we found that there are 333 genes that were found to be in common between inoculated samples and the mock ones for these two groups. And we can also see here that these genes were mainly associated with defense responses and immune system process. And we can also see that genes were associated with response to chitin or regulation of defense response to fungus. And this denote the activation of a PTI response by the recognition of the pathogen's conserved features. Then the enrichment of GOT terms associated with cell wall thickening by callus depositions, for example, indicated a response with callus deposition in both type of samples. So in the literature, it is known that RW presents a resistance to powdery mildew, and this resistance is associated with papilla formation. But also, our microscopic observations show that there is a callus depositions in response to black spot disease infection as well. So we hypothesized that the response highlighted here are either a result of the presence of powdery mildew in both type of samples, or that RW activates the same type of response against both powdery mildew and black spots. It is so. These are hypotheses that need to be studied further. So this diagram here represents an overview of the results found during the analysis of all the groups for the time course analysis. And we combine these results with the macroscopic studies that we carried out in part 2. And we can hypothesize that the resistance of RW in old leaves is based on 2 mechanisms. So first, the chitin recognition that leads to an activation of the PTI response. And this is characterized by a callus deposition that was observed at the macroscopic level. And then we also hypothesize that there is a recognition of effector that activates an ETI response. And this is characterized by an early HR response that is observed in old leaves of Allbush. So finally, we also observed the activation of defense pathways associated with defense response and localized cell death. And so in light of these differentially expressed genes, we hypothesized that the partial resistance of RW involves both a PTI and an ETI response. The existence of the ETI response suggests that the partial resistance of RW might be based on our genes. However, the results obtained in this part remain untested as the contamination of the mock samples was observed, and we cannot rule out the existence of an interaction between both fungus. Therefore, further experiments need to be carried out. So to conclude, the partial resistance of RW is a complex incompatible interaction with 2 main characteristics. So it is a typical quantitative trait loci controlled by several stable genes, and also a complex incompatible interaction. So first, we saw that there are 2 main QTLs that were detected, and we were able to reduce the QTL confidence interval to 10 centimorgans. Then we saw that there is maybe more than 2 QTLs for B3 and B5 with the calculation of the meta QTLs, but we are still unsure and need to do more tests to verify this hypothesis. Then, as the resistance of RW was tested in several locations in a natural infection, we don't know which strains are present in our fields, so we hypothesize that maybe partial resistance of RW can be broad spectrum. Then we also saw that these main QTLs have different effect on the phenotype, with LG3 having an effect on the penetrance and the severity, and LG5 only the penetrance. Finally, it seems that partial resistance of RW can also have some background dependent QTLs detected on B4 and B6. So concerning the interaction type between the rows and the pathogen, we can see that there is a leaf age effect for which the resistance effectiveness is a function of. Then we observed, we didn't present that today, but we observed that the cuticle importance the cuticle is very important in the resistance, and we want to study that further. Then in old leaves, we saw that there is a PTI immunity, that a PTI response that is activated that leads to a callus deposition, and also an ETI response that leads to early hypersensitive response. So now what is left to know is how do the loci that we detected on one side participate to the partial resistance of RW. And so here I would like to present you some various perspectives because these findings are the starting point of many investigations of black spot disease in our team. So quickly, we used several QTL mapping, and so we can increase the power of the QTL detection and narrow down the location by using different scoring scale or components of partial resistance assessment, but also increasing the population size. So this has been done already. And we are also starting a fine mapping using a set of SNPs that are in the meta-QTL region and that we are transforming into CASP markers. Then to study the importance of the cuticular barrier, we can quantify the infection in leaves with and without a compromised cuticle. And we have also studied infiltration assay with Professor Debner to avoid cuticular resistance and study post-penetration resistance. Then to study further the leaf age effect, we can wonder if it's a matter of time or quantities. And so we can quantify the cell death or the production of ROS with DAP staining, but also the callus concentration over the time. Finally, in order to link these two results, we need to proceed with further characterization of each QTL. And for this, we have selected individuals in the OW population that presents resistant alleles in the QTL locations, and we have been tested different strains on them. Finally, for the transcriptomic data, we need to present some qPCR validation of the RNA-seq data, and also to study the expression of these genes in samples that do not have a purge mildew infection, so we can validate or not our hypothesis. And finally, to solve maybe the problem of biological replicate variability, we can separate the biological replicates by so separating them and using specific methods and algorithms to detect them. So I would like to thank the funding structures, so the BAP and the region, but also the RFI for the mobility that I was able to carry out in the Professor Debner's lab in Germany. Then I would like to thank the experimental unit and Phenotic for the care of our plant in the field as well as the greenhouse, but also the EcoFen team with Marie-Noëlle Bélanger that helped me with the propagation of our strain. And I would like to thank the platforms of the SFR CASAV with Anan. Thank you, Muriel, for your help in the lab, and the IMAC platform, so Aurélia Roland and Fabienne Simoneau, that helped me to carry out all the microscopic studies and to take these beautiful pictures. And then I would like to thank Professor Debner and his team that I see are almost all here. So thank you all for welcoming me there, and Julia for your tremendous help when I was there, and Yanis and Helena for introducing me to the team. And then thank you for my CSI members and for my Agrinium professional referee. So I am part of the 7th promotion of the International Research Agrinium, and so I would like to thank them all for all these good times and discussions we had about agriculture. And of course, my GDO team. Thank you, Tatiana and Jérôme, for the help taking care of our plant, and Annie and Julien for your help in the lab. And of course, all the people that I met in the IRHS and with who I spent really, really good time eating and talking. And finally, last but not the least important, Fabrice, my PhD director, and Laurence, Vanessa, and Sophie, thank you for this really good talk, laugh, and nice meetings that we had. And thank you for all my friends and colleagues, because 3 years of PhD without you would have been the same. And thank you for my family and my fiance. Thank you all. So.